I would like to introduce our first speaker, Dr. Sarah Berry. Sarah is a reader in nutritional sciences at the King's College London and is also Chief Scientific Officer at Zoe Ltd. Her research interests relate to the influence of dietary components on cardiometabolic disease risk, with particular focus on personalized nutrition, postprandial lipid metabolism, and food and fat structure. Since commencing her research career at King's College, she has been an academic leader for more than 30 human nutrition studies in cardiometabolic health. She's leading the PREDICT program of research assessing the genetic, metabolic, metagenomic, and meal-dependent effects on metabolic responses to food. So this research is at the forefront of developments in personalized nutrition and is forging a new way forward in the design and implementation of large-scale remote nutrition research studies integrating novel technologies, citizen science, and artificial intelligence. So Sarah's Zoe Predict research and its application has significantly contributed to healthcare and research innovation. With this, please, Sarah, give your presentation on personalizing diets from genetics to behavior. Where do we stand today?
Thank you. I think actually you've just summarised my whole talk there, so we can move on. So firstly, I do need to declare conflict of interest that, as Petra said, I am also Chief Scientist at Zoe, who are a personalised nutrition company. But let's move very quickly into the content of my talk. I'm actually going to give quite a broad perspective. I am giving a couple of talks on Thursday, so if you want to hear a bit of a deeper dive into some of the complexities, do come along to those symposium talk. And I am going to be presenting some new results after this in an abstract session as well. So this is really to set the scene very much for the next two talks and to tell you really about my perspective of where I think we are in terms of personalised nutrition. And I think we need to be asking ourselves not just how we can transition from the average to individual advice, but actually, can we? Is this possible? And is this possible given how complicated we are? How complicated we are as individuals, how complicated the food is that we eat, where we now know food isn't just a simple list of macronutrients, how complicated how we eat is. So whether it's the meal order, how fast we eat our food, whether it's how much we've fasted, how much sleep we've had, and also the complexity of what drives some of the behaviour around why we make the choices that we make. And so given all of this complexity and given the fact that actually there's really robust evidence that underpins population-based guidance, should we even bother delivering personalised dietary advice? And so I think we need to really take a step back and say, well, why do we need personalised approaches? And the way I see it is it's for two reasons. It's firstly to improve the efficacy of the advice, given our growing understanding of how we all respond so differently to dietary advice, to lifestyle, etc. But also given that actually less than 1% of people in the UK, and the same applies in many other different countries, actually adhere to population-based advice. And I think we also need to think, when we're thinking about personalised nutrition, that it's not just about who we are. So I think there's this very kind of rigid structure or rigid way of thinking about personalising based on our biology, base it on our microbiome or our genetics or on using a continuous glucose monitor. I think we need to think about all the different features that we can personalise when we think about the practical application of personalised nutrition. So this is in addition to who we are as individuals and all that complexity I showed in the previous slide. It's also personalising based on where we are now. What are we actually eating now? How far do we need to move the needle? What about how we eat? So those factors I mentioned earlier. What about also why we make those choices? And what about our goals? What are we motivated by? What do we want the end target to be of any dietary changes we make? And what about how we deliver that advice? Whether we deliver it through an app, whether we deliver it in a really intensive way, for example. We need to consider all of these factors when we think about personalised nutrition. Now, it wasn't possible, I don't think, to consider all of the complexity of who we are, of our food, of how we live our life, why we make the choices we do, and also these kind of personalisable features that I talked about in terms of delivery as well previously. And that's because previously we've always had to rely either on generating data at really large scale but low precision, so for example from epidemiological studies, or from generating data that's very high precision but very low scale from traditional randomized controlled trials. But we're at a really exciting time in nutritional research where we have these big data opportunities, where we can collect data at the scale, breadth, depth and precision that we need to make personalised nutrition a reality. And this is because we can leverage opportunities such as digital devices, and I'm sure everyone in the room here has some sort of device that can track something related to their health or their diet. We can leverage clinical devices such as continuous glucose monitors. We can leverage remote clinical testing such as DNA, saliva swabs, or microbiome samples from our poo. So we can actually, from the comfort of our armchair, take all of these tests. And we can also leverage citizen science or population-based science, whereby people are interested in finding out more about themselves and then can share the data back with us. And this enables us to transition from this general population-based advice to personalised advice. But we mustn't forget what I think is a really sweet spot in the middle, and we're going to hear about a lot more in the next talk, this area of stratified nutrition, where we can group people based on shared characteristics. And I think we mustn't jump straight to thinking it's either population or personalised based advice. I think there's a real magic in this area of stratified nutrition as well. Ultimately, both stratified and personalised nutrition will improve adherence and will improve efficacy, in my opinion. And what this will enable us to do is then transition from this average population-based advice to this individual-based advice in a way that simply wasn't possible, say, 10 or 20 years ago. And this is exactly what we're doing with our PREDICT programme of research. This is a huge ongoing programme of research which is leveraging remote technologies, remote clinical trials, and it's a collaboration between Zoe, who are a tech healthcare company, and also very traditional academics like myself. And we're collecting data at a rate of about 4,000 people a week, and so we have data now on about 150,000 people, and we're just continuing to collect more and more data. And we're using this data to explore three key questions, to look at how much variability there is between individuals, but also, really importantly, within an individual. And this is something that I'm going to focus on a little bit today, because I think it's something that isn't given enough airtime in the field of personalised nutrition. We're also trying to look at what's explaining the variability in responses, again, between individuals, but also, importantly, within an individual. And also, can we predict how an individual is going to respond? We need to be able to predict this in order to then offer that personalised advice. The way we're doing this is using the kind of remote technologies that I mentioned earlier, using remote clinical testing. We have an app, and this app allows us to collect really high-resolution diet data. We use different means for dietary assessment. We use weighed records, barcoding, image records, but we collect diet data right down to the brand level. So that enables us to look at even what type of emulsifier or additive is in that food, never mind just what the macronutrient composition is. We collect daily data on mood, energy, hunger, and so much more each day that they're using the app. We also provide people with nutritionally matched test meals so that we can look at, again, between and also within person variability. These are given in duplicate using a standardized kind of baseline. And then we fit people with wearables. We fit people with activity monitors so we can look at sleep and physical activity. We fit people with continuous glucose monitors so we can look at 2-week glucose changes. We also use dry blood spot technology to look at lipids and metabolomics, and we collect stool samples as well for microbiome testing. So what can we learn from the data that we've collected? Well, I'm going to focus mainly on our first study, which was our PREDICT-1 study. Now, this is a study in 1,000 people in the UK and 100 people in the US. It's a little bit different to all of our other research in that it did actually also involve a very tightly controlled clinic day where we looked at postprandial measures over 6 hours in the clinic. But it's the data and the learnings from this that have gone on to inform us for all of our future research. So what I'm going to show you here first is the mean response. And this is the mean response from our clinic tests where people were cannulated, and we collected every 15 minutes after a high-fat and a high-carbohydrate meal blood samples to look at all sorts of metabolites. And I'm going to just show you triglycerides and glucose for the rest of the presentation, just to keep it simple, because they're there to illustrate a point. So what you can see is on the left, you've got the postprandial triglyceride response. That's up to 6 hours after having a high-fat, high-carb breakfast, high-fat, high-carb lunch of 4 hours. And then on the right, you've got the glucose response, which again you can see after the high-fat and high-carb breakfast and high-fat, high-carb lunch. Now, if I overlay each and every individual's response, this is the 1,000 individuals in the UK, you can see just how variable people's responses are. Now, I know that everyone in this room is probably aware that, or is aware how variable our responses are to food, but I think this really nicely illustrates just how meaningless for some of these people that have this really high triglyceride response or really low response, the mean is for them. We've also quantified the size of this variability, both between individuals, but we're also able to quantify the size of the variability within individuals because all of our meals in Predict1 were given in duplicate. And so what you can see if we use triglycerides as an example, that the between person, the inter-individual variability is about 70%, and the intra, the within person, that day-to-day variability is a lot lower at, I think, 30, I can't see, it's a bit small, sorry, at 36%, so around 40%. But that's still really high, and this is why it's really important we consider that day-to-day variability, whether it's measurement error in the tools that we're using or whether it's true day-to-day variability in how we respond to food. So are all these differences in our genes? Well, I was a child growing up in the 70s and I was told it's all due to your genes, it's the age of genetic discovery, there's nothing I can do about it. Thankfully, we now know that this isn't the case, and I think this is also nicely illustrated here. So 70% of our population from Predict1 were from the Twins UK cohort. So we had a large proportion of identical and non-identical twins. And what I've done here, just to illustrate how much genetics does or doesn't contribute, is plot one identical twin against another identical twin. Now, if you look at the figure on the left for height, if the trait is all due to genetics, which it predominantly is for height, you'll get this beautiful, nice straight line like you see for height when you plot one identical against another. When we do this for glucose, and this is the postprandial glucose response that I showed you in the previous slide, and we do it for triglycerides, again, the postprandial triglyceride response in the previous slide, the dots are all over the place. Okay, so it's not all due to genetics, and we can even quantify this using something called ACE heritability modelling because we had our identical and non-identical twins. And we can see if we use ACE heritability modelling that less than 50% of that variance that we saw before is due to genetics. So genetics aren't explaining most of the differences. So what is explaining all of the differences? Well, with the kind of data sets that we have, we've been looking at the multiple exposures, responses, and outcomes. And I don't have time to go into all of that today. I'm going to pick up on a couple of these, which are going to be the microbiome and also the area that I'm most interested in at the moment about how we eat. But what I've done here is illustrate how much variance is explained by different factors. And what you can see is that for glucose, for example, the composition of the meal, genetics, and how we eat is really important. But actually for postprandial triglycerides, it's less important. And we've done this for lots of other outcomes as well. And why is this important? It's important because we need to, as nutritionists, think about what outcome we're interested in terms of where we put most of the effort for the advice related to the exposure that we want to modify. Okay, I can't talk about personalized nutrition without talking about microbiome. I'm not a microbiome expert. I do always have to caveat that when I cover these couple of slides. We are doing a lot of work at Zoe and a lot of work with our Predict studies on the microbiome, so I just want to touch on it for a moment. We've done a real deep dive, not in just our Predict1, but also our Predict2 and Predict3 studies. And what we've identified is what we're calling a microbiome signature. So a signature of species that are associated with healthy measures of diet and favourable measures of health. This was published in Nature Medicine a couple of years ago if you do want to dive into the details. And we're loosely calling this panel a bugs, and I know some microbiologists may hate this term, so apologies, but we call the ones that are associated with favourable measures of health and favourable measures of diet are good bugs, and those conversely associated with unfavourable diet and unfavourable health are bad bugs. This is all association data, though. So can we actually use diet to change the microbiome to elicit a change in health outcomes? And this is something we've been looking at in our current Predict3 study, where we've had about 20,000 people retesting their microbiome after we've given them dietary advice where microbiome is one of the input variables guiding that advice. And yes, we do see a change in the microbiome. We do see an increase in these good bugs, and we do see a reduction in bad bugs. And then what I'm going to be presenting in about an hour and a half as an oral abstract is the results from our randomized controlled trial called our method study. And it's the first time we're presenting the results where we also looked at the microbiome in the people following the Zoe personalised programme. Okay, another area that we've really delved into is this area of how we eat. And this is something I'm particularly interested in. This is because how we eat underpins a lot of this day-to-day variability that we see. So that day-to-day variability that I talked about earlier, that 36% in variability in postprandial triglyceride response, for example. And it's really important that we look more at this as well as the biological variability. And I'm going to have just one slide that summarises about four different areas that we've been looking at. So sleep. We know from our own research, and this was published a couple of years ago in Diabetology, that if you have a poor night's sleep, your postprandial glucose response the next day to a standard meal is worse, i.e. it's higher than if you have a good night's sleep. So if I've had a good night's sleep and I have exactly the same breakfast one day, I will have a lower glycemic response than if I have a bad night's sleep the next day. We know time of day is important. Now, this isn't anything new. We know that there's some great trials that other scientists have published showing that there is a lower glycemic response to a standardized meal if you have it in the afternoon compared to if you have it in the morning. But we want to look always, does this apply to everyone? Does everyone respond in the same way? And so this is a great example of where, again, we can just look not even in a personalised way, but in a stratified way. So when we stratified people according to their age, which is what these bottom bars are, and I'm sorry if the font is a little small, but on the left side are those that are under 40, and on the right side it's going up in decade increments. So what you can see is if you're yet less than 40 years old, yeah, time of day is really important. We might need to think about what time of day we're having our carbohydrates. Once you get older, it doesn't really matter that much. Now, it could just be because they're less insulin sensitive when they're older, but how does this matter in terms of the advice that we're giving? Do we want to give the 70-year-old the same advice that we're giving the 40-year-old? We've also been looking at meal order, and again, this just illustrates the complexity of delivering personalised advice. And this was a sub-study that we did in 100 people from our Predict 1 study, who we invited back to do another 2-week study called our Predict Carb study. And what we did is we randomly allocated people to starchy staples. We gave them at breakfast, and then we gave everyone exactly the same lunch. They all, each day in a crossover fashion, had different breakfasts. And what we found was that one of the primary drivers of their glycemic response to lunch was what they had had that morning for breakfast. So in this case, we found that people that had mashed potato, and I know that very few of you probably have a big bowl of mashed potato, or I hope you don't for breakfast, that people that had mashed potato had this really big glycemic response to a lunch meal, yet if they had the other starchy staples, they had a lower response. Something else that we started looking into in collaboration with Kieran Ford is eating speed. And again, this is just a really nice illustration of a practical way that we can actually personalise advice. What we know from our data is that people that eat their food more quickly have a higher adiposity, they have a higher waist circumference, higher BMI, and a higher energy intake. And they also have a higher 15-minute postprandial glycemic response. So even regardless of their genetics, their microbiome, their metabotype, we could actually just say to someone, hey, how fast do you eat your food? Well, put your knife and fork down between each mouthful, slow it down, and we can personalise advice in that way. And I think this is where the real magic of personalisation lies. Thank you. Okay, so something I wanted to touch on as well is continuous glucose monitoring. Anyone who is on Twitter or X will know it's very contentious, that you're either against it or for it, and it's an area of great controversy. Should we be using continuous glucose monitors in healthy people? We are, in the work that we're doing at Predict, but so are many, many other companies, and so are many people using continuous glucose monitors. So rather than fight about whether we should or we shouldn't, let's learn from the data that we're collecting in healthy people, and yet let's use it well. And I want to just give you one slide of what I think is a really great example of how, again, we can personalise on features, but also use a measurement tool like a continuous glucose monitor well. So the response that we get when we consume carbohydrates throughout the day is complicated. The response curve throughout the day is complicated. Up here is the kind of outcomes we get from our continuous glucose monitor. It is cartoon style, so it doesn't show all the minor oscillations. And in the cartoon style, I've shown what is, I would consider, a more unfavourable response in red and a more favourable response in blue. So we get lots of metrics. We get things like time in range, the area under the curve, the peak, the dip, the variability. We know that all of these different metrics are differentially associated with disease. So we know that time in range is important for insulin sensitivity. We know peak is important for inflammation. Something that hasn't been looked at much is glucose dips. And this is an area that we've spent a few years delving into. And what we did is in our cohort, we separated people out according to whether they were big dippers or little dippers. So 2 to 4 hours after having a high carbohydrate meal, we found that about 25% of the population had these big dips. And what we found is that people that were big dippers had an increase in hunger. They had an increase in energy intake at their next meal of 80 calories, and over the whole 24 hours of 320 calories. They felt less alert, and they had less energy as well. Now, these people would not have been identified using traditional measures such as the 2-hour area under the curve. But this is between people. What about within people? And so within our Predict2 and our Predict3 cohort, we've started to look, what about between a person? If one day for breakfast they have a dip and the next day they have a breakfast and they don't have a dip, does it matter? And we see, yes, it matters. And what we see is within a person, if they have a dipping day, they go on to have a reduction in energy, increase in hunger, and they have about 160 calories more on average at their next meal. So this just demonstrates that complexity. So to finish up, what do I think is the future of personalised nutrition? Well, I think something really important that we need to move away from is thinking that personalised nutrition has to be at the exclusion of population-based guidelines. It can be a beautiful overlay. There's a real place for population-based guidance, and I've tried to illustrate this here from one of my studies. So you can see from the figure on the left that everyone is responding the same way. So each of these lines is an individual's response. In this case, where everyone responds in the same way, we should be giving everyone the same advice. So the same for fibre, for example. Yet in the figure on the right, you can see that actually about 60% favour the intervention, about 40% favour the control. In this case, maybe we want to give different advice. And so I think it's a great example of how personalised nutrition can be an overlay to this population-based advice. And lastly, does personalised nutrition work? Well, we know that personalised nutrition improves adherence. There's many, many studies. The Food for Me is just one study showing that personalised approaches improve adherence. But does it improve people's health? Is it more efficacious? And there's the application work from the original work that Erin Segal did that was published in Cell, which developed a personalised nutrition approach where they saw when they used this over I think that's 6 months, 6 months, a reduction in many different health outcomes that you can see here, weight, triglycerides. And there's also the Preventomics body of work that we're eagerly waiting publication, two of the studies of which have shown an improvement in outcomes, but one that hasn't. I do have the results from our Zoe RCT that I'll be presenting in about an hour that integrates all of these different features that I don't think these other studies are really tackling. So what's next? Well, I think what's next is that we need to think about this day-to-day variability. We need to be thinking about collecting real-time data that's driving this day-to-day variability. We need to also be thinking about how we go about affecting change in people once we've got all this information, and how we can actually make this accessible to everyone at a population level and where population-based guidelines fit in. But the most important thing, and I do finish all my talks saying this, so apologies if any of you have been in my talks before, is something that we mustn't forget is the pleasure of food. We don't want to be sitting at a dinner table where, I certainly don't want to, my husband's having his personalised meal, I've got my personalised meal, my son's got his, my daughter's got hers. I want us to eat as a family and enjoy our food together as part of our cultures. It's linked to our emotions as part of our lifestyle. So I think it's really important we remember that when we think about personalised nutrition. And thank you to the great team of people that I've worked with at King's and Zoe that really are the ones that do all the hard work of what I presented today. Thank you.